Good morning, everybody. It's my great pleasure to present our R&D progress of PMC-43 at this SCW Ophthalmology Virtual Conference. As I was introduced, I'm Jin Sun-Yu and CEO of Pharmapsin. We are clinical stage biotech listed at Kosdaq stock market in South Korea. So the contents of my talk, I would like to introduce Tie2 biology in ophthalmology. And then I would like to happy to share what the PMC-43 is. Actually, this is Tie2 targeting and activating fully human IgG. And we are developing in both ophthalmology and oncology. Then I will close my talk with our future plan. So Tie2 stands for tyrosine kinase with Ig and EGF homology domains too, and it expresses on endothelial cells and a subset of macrophage. This receptor possesses a unique extracellular domain containing two immunoglobulin-like loops separated by three epidermal growth factor-like repeats that are connected to three fibronectin type 3-like repeats. In cancer patients, Tie2 is expressed in a subpopulation of monocytes that home on the tumor and are essential for the formation of new blood vessels. The ligand for the receptor is angiopoietin-1 and angiopoietin-2 so far. Tie2 has also been suggested as a marker for nucleus purphosus progenitor cells from the intervertebral discs, which upon activation of angiopoietin-1 start to multiply and differentiate. Hereditary inactivating mutations in Tie2 leads to primary congenital glaucoma. Defects in Tie2 are associated with inherited venous malformations. The Tie2 signaling pathway appears to be critical for endothelial cell smooth muscle cell communication in venous morphogenesis. Disease-associated dysregulation of circulating Tie2 ligands contribute to pathophysiology and patient outcome. The NG1, NG2 Tie2 signaling in vascular endothelial cells, we can sum up as NG1 as a activator and promotes endothelial cell survival and vessel stability and barrier function via activation of a signaling receptor Tie2. And its downstream target is PI3K, AKT, and Rho family small GTPases. And NG1 Tie2 PI3K, AKT signaling inhibits NF-kappa B, FOXO1, and FOXO1 induced NG2. And NG2 is a weak agonist or antagonist which modulate NG1 Tie2 pathway. Often inflammation and vascular injury, NG2 stored valvular bodies are released in circulation and antagonize NG1 Tie2 signaling and then increases FOXO1 and NG2. Unlike signaling receptor Tie2, Tie1 is an orphan receptor homology of Tie2 and mostly does not react to ligands. In some cases, Tie2 signaling is stimulated by phosphorylation due to in part of the Tie receptor complex is Tie1. Tie2 activation is also achieved by the inhibition of VPTP, which dephosphorylates and deactivate Tie2. But I have to tell you that still we know just fraction of information in angiopoietin and Tie2 biology so far compared to VEGF-2 signaling cascade. So NG2 Tie2 leads dysfunctional vasculature. This is well known. So as you can see on left upper panel shows transmission electron microscope image with the schematic indication of vascular endothelials and pericyte. And the right upper panel indicate that abundant angiopoietin-1 enhances endothelial barrier function and stabilize vessels. In contrast, abundant angiopoietin 2 decreases endothelial barrier function and increase vascular permeability, leading to the formation of leaky, disorganized vessels, as you can see on right lower panel. This slide shows dysfunctional angiogenesis and pathological vasculature in age. So when we are getting older, our vasculature too, and there will be more dysfunctional angiogenesis compared to when we are young. And we are getting older, when we are getting older, the density and type 2 copy numbers, intensity decrease in our choriocapillaries. And the target of our PMC43, which is type 2, plays important role for choroidal blood flow as published. So based on this limited scientific rationale, we have isolated anti-type 2 agonizing antibody collections. And I would like to introduce one of them, which is PMC43 for today. So what is PMC43? So in pathological conditions like diabetic macular edema, diabetic retinopathy, and age-related macular degeneration, in that environment, VEGF and also angiopoietin 2 are highly overexpressed and cause vessel leakage, which leads to vision impairment and ultimately blindness. In normal condition, angio-1 activate type 2 receptor to maintain the homeostasis of vasculature in the eye. But in the pathological condition, we need a much stronger type 2 activator, and we strongly believe PMC43 can fulfill that mission. So PMC43 as a vessel normalizer, so it has the ability to change disorganized pathological leaky vessel to healthy normal vessel. We can apply for various disease indications, including not only ophthalmology or oncology, but many others in our bodies. Currently, marketed therapeutics for age-related macular degeneration or DR, they're all similar mode of action, which is anti-VEGF. And these drugs are selling multi-billion dollars, but also effective to block the VEGF biology in that eye environment and retard the disease progression. That's the fact. But it induces transient vascular normalization. That might be kind of limitation, we believe. And high dose and long-term therapy leads massive vessel pruning, resulting in immunosuppression. And it also linked to limited improvement of visual acuity in over 30% of all AMD, DME, and DR patients. And interestingly, approximately 30% of patients, they do not respond to anti-VEGF therapeutics. And most anti-VEGF therapeutics require, so far, intravitreal administration at intervals for each month. So every month, the patient should get the injection. Well, now there is some improvement so that every 8 weeks can be done, but still in this COVID-19 pandemic, this is kind of discomfort for patients. So we need a solution for this, and we strongly believe inducing active vascular stabilization by type 2 biology mediated therapeutics. And that way we can enhance the clinical benefit through steady vascular normalization and enhance long-term vessel regeneration that contribute increased intervals of drug administration. This slide shows the competitive landscape. So if you look at the left panel, Lucentis, Eylea, Avastin, Beovu, they're all marketed drugs. And very recently, Roche's bispecific antibody faricimab is FDA approved. But again, these are all anti-VEGFs. And on the right, there's Unity Biotechnology is developing UBX2050, which is tied to agonizing antibody like Parmapsin's PMC43. And there's a company called Vasubune has a vascular tie. This is peptide, multimer binding type 2 and agonizing type 2 downstream signaling. And there's AOP Therapeutics. They have both small molecule and antibody targeting VPTP and blocking the VPTP, and that lead to type 2 activation. And then there are some other assets too. So this slide shows where PMC43 stands. It binds directly type 2 and then activate the downstream signaling of type 2. So I will shift the gear to introduce our in vitro data of PMC43. So PMC43 was isolated by fully human antibody called HuFace Display Library, what Parmapsin in-house developed, an antigen customized selection system. And this PMC43 binds both murine type 2 and human type 2 with nanomolar affinity. It was confirmed by FACS, but also BIAC1. And PMC43 activate human vasculature by using HuBXL systems, so you can see. And it also ubiquitinated the FOX1 proteins by Western blot and also in these assay systems, regardless whether it is NG2 present with PMC43 or PMC43 only, suggesting that PMC43 is quite competitive regardless whether NG2 is present or not. And PMC43 also inhibit VEGF-induced vessel leakage. So in this study has also shown that in the presence of VEGF, which lead to vessel leakage, but together with PMC43, protect the vessel leakage better than NG1 actually. In the right panel, you can see. And also PMC43 recovers cell-to-cell tight junction as a vascular endothelial criterion. So usually VEGF induce the V-criterion phosphorylation, and in the presence of PMC43, it can be inhibited too. PMC43 is IgG1 subtype, so it might have potential effector function, but fortunately, this antibody has no effector function and proliferation or no T-cell activation effect, which is desirable for ophthalmology purpose. And PMC, interestingly, we have to figure that out why, but PMC43 also reduce the Ty1 expression level. So I'm going to shift to in vivo study of PMC43. We used murine CMV animal model system, and we perform side-by-side with Eylea. And as you can see, both Eylea and PMC43 has a similar effect to reduce the vessel leakage area. However, we are quite excited about in ERG test comparison. And in ERG, our PMC43 has a better performance compared to Eylea. And when we moved to from murine to monkey CMV model system. Like murine CMV model system, PMC43 and ILEA have approximately similar performance in terms of vascular leakage reduction. However, in terms of ERG test, PMC43 has a better performance. But I have to tell you that this has a limited number of cases, so we are planning to expand this study too. And also in monkey CMV model system, we look at the comparison of ILEA and PMC43 in angiogenesis in terms of VEGF and NG2, and also vessel normalization by indication of PDGFR, and also inflammation in COX-2, which is similar range. And this is still ongoing studies, but we have a preliminary data that PMC43 has a biology also in DR in DBDB mice systems. So in our plan, we are gathering IND package data so far so that we can file our IND in Q3 of 2022. And we're gonna recruit patients from wet AMD and DR, DME, and RVO who were under the treatment of ILEA or Lucentis or Avastin. And we will determine the dose escalation, MTD, and kind of efficacy too. So in summary, again, PMC43 has a nanomolar affinity to human and murine type 2, and also it has a increased biology of endothelial migration and prevent its permeability induced by NG2, and PMC43 inhibit VEGFR2 activation induced by VEGF, and also inhibit vascular leakage in mouse CMV model system, and inhibit laser-induced neovascularization in both murine and monkey CMV models. But exciting to observe that in ERG test in monkey and in mouse system, PMC43 has a better performance compared to ILEA, and we are preparing for our IND for next year phase 1. So this is my contact email address. We are open for constructive research collaboration, and thank you very much.